So I'm Paul Denny. I'm one of the curators at UniProt, and I'm going to take you through an introduction to using the UniProt website. And also helping me today is one of the other curators, one of the senior curators, Rosanna Zaru. So what I'll do is I'll talk you through an overall guide to using the website to look for proteins, to look for data, for function associated with those proteins, features like sequence, and then within a sequence, different parts, different regions of a protein, post-translational modifications and other kinds of amino acid modifications. Proteomes, I'll briefly sketch a little about how UniProt gives you access to the complete set of proteins from a particular organism. And then finally say a little about annotations for mutations and disease that are relevant to proteins in the UniProt database. So what is UniProt? It's also known as the Universal Protein Resource, and it's the result of efforts between a big consortium of 3 main organizations, bioinformatics databases at institutes in the UK, Switzerland, and the US. And together they provide to the scientific community, it's all freely available, freely accessible, a comprehensive and high quality set of data resources about protein sequence and function. And it includes things like pathway data, interactions, subcellular location, the actual primary sequences themselves, isoforms of proteins, disease variants, and modifications. So these are the core principles on which our website is built. You should be able to find data about proteins. You should be able to download that data in multiple forms. And then you should be able to visualize, make sense of those data. So there are ways to look at the whole protein or particular regions, and other kinds of information such as a 3D structure viewer. And then there are analysis tools associated with the website. You can look for similar proteins using tools like BLAST. And then once you've found similar proteins, align them with one another. Or if you want, so let's say you've got a short peptide and you want to find proteins that contain that peptide, you can do that. And one of the other key parts of the database is being able to find identifiers, unique identifiers, or even redundant ones built into the database. And then once you've got to a particular protein that you're interested in, you can find out more about those proteins. And it's very flexible. It's quite customizable. You can choose which particular kinds of data you want to view or to download. Where does all the data come from? It's primarily from the International Nucleotide Sequence Consortium, or the INSDC, which you can see here. So it consists of 3 organizations: the database of nucleic acid sequences in Japan, in the US, and in Europe. But other data is pulled in from other sources. There are direct submissions to UniProt. People send their sequences to us. And then there's data pulled in from the Ensemble genome browser and database. And then, of course, data comes in from the structure databases like PDB. So this gives you an idea of what you see when you get to our homepage. Up on the top left here, you can see the tools that are associated with the website. And then this box here highlights the different resources within the database and on the website. And then in the top right corner here, this gives you the details of the latest release. Our data is released on a approximately 8-weekly cycle. And the current one, I think this is actually now on 04 for 2022. So this slide is actually already slightly out of date. And then these little icons here are for other tools that are useful for accessing the data and your results. So here's the search bar, which may look quite familiar to you. It was deliberately designed to be a bit like many other search tools. And there's a drop-down menu on the left here. You can look at what's called UniProtKB, and I'll say a bit more about that in a minute. And then you can type in here all kinds of, in the search bar, all kinds of information, a gene name, a protein name, a disease. There's a lot of different, well, some examples are shown here of the kind of thing that you could enter into the search bar. And then over here on the right, there's a couple of more advanced search tools. There's one which allows you to construct a query, which I'll show you later on, using a combination of terms. And then list refers to ways to take multiple IDs or accessions or information about proteins, a whole set of proteins, and put them in at once. So I'm going to start off by giving you a few examples. So let's imagine that you're interested in a particular biological process. It just so happens that these proteins in this image are all involved in the process of recycling in the cell, of autophagy or autophagy. And I've shown here in this diagram the gene names for a couple of the proteins, actually 3 different proteins involved in this process. So you could either search individually for each of these in the search bar that I just showed you, or you could enter multiple ones and you can then filter the results. So this gives you an idea of the advanced search. You can choose first the core database that you're searching, and then how you link the different terms together. So do you want to have them linked using the logical relationship and, or, not, and so on. And you can choose to restrict the taxonomy, for example, and then you can keep on adding more fields. So if you wanted to add more, you click on the Add Field button, and you can also remove some. Let's say you want to look for multiple proteins at once. There are 2 ways you can do this. You can either use this button up here at the top called ID mapping, or you can go to the list at the end of the search bar. So these are 3 of the identifiers that I just showed you in one of those previous slides. I've got a list of gene names, and I've decided I'd like to search for these and put them into this search box. So I've got a list of gene names, and then I'm deciding to restrict this by human. So I'm only interested in human proteins. And this is an example of the kind of thing you might get as a result. Now, one of the things about the tools is you can keep this data. So it's personalized to your browser window. So even if you go away and leave this and come back to it, it stays there, and it lasts for 7 days. If you look across the top here, it mentions these results are here for 7 days. In fact, they're there slightly longer than that. They're there for 14 days, but you have to rerun them if you want to see the results when they've been there for longer than 7 days. The one at the top is just the one you've done most recently, and then further down the page, you'll see previous searches that you've done. Now, once you've got the set of results, let's imagine that we've done a search and we've got these results. You can choose to filter them. So down the left-hand side here of this search, this bar on the window, you can decide whether you want to get rid of a filter, add a filter. So let's say you're only interested in things with 3D structure. You could choose to just view those. And one thing that the eagle-eyed amongst you may have noticed is in this result, we've got a little icon here that's gold or a kind of silver gray. Now, these I'm going to say a bit more about in a minute, about the difference between these kinds of icons. So there are two overall classes of protein, or protein entries rather, in UniProt. There are those that have been reviewed, so they're sometimes referred to as the SwissProt set, and there are those that are unreviewed or tremble. And the difference between the two, the reviewed ones down the left-hand side here, these are protein sequence entries which have been manually curated. They've had data added to them by a human curator. They've been reviewed by those people, and they've gone through the literature and added experimentally verified information. The other set, the unreviewed tremble entries, they've also been analysed, but they have been computationally analysed. So they have annotations that have been created by a pipeline of tools automatically. And so there's a continual process within UniProt of curators taking unreviewed entries and then reviewing them, and then also updating the data for the reviewed entries. So if we went back to that previous slide, this gold little icon here, this is an entry that's been reviewed by a curator, whereas the silver grey ones have not. They're tremble entries. They've been automatically annotated, but not touched by a human curator. Going back to our identifiers, in this example, instead of the gene names, I've got a set of UniProt identifiers. These are accession numbers instead. You may find that you've got a paper or some other source which only gives you these kinds of identifiers. It doesn't tell you what the gene names are. And this means that you can use the same tool to now convert those if you want to know more about other kinds of information, such as the gene name. So if I paste in those identifiers, I can then find out the gene names. And in this example, you can see I've just converted back to the gene names that were in one of the earlier slides. So this first one here is ATG12 and so on. One other thing which users occasionally want to do is that they've got a short peptide sequence from whatever source, and they want to know what entries are there in the database that contain this sequence. So this is called a peptide search. And this is an example of the kind of thing you might get as a result. We've got 2 reviewed entries and 2 unreviewed tremble entries, and you can see their icons here. If we use this peptide shown in that previous slide. Moving on now to a different example, we want to find something out functional about these protein entries. So if I take IKK as a search term, I would get a very long list of proteins. I mean, I think, yeah, there's over 200 here. And I want to know, let's say, for example, I want to know about the human IKKA, also known as CHUK, C-H-U-K. That's its gene name or gene symbol. If I click on that link for that entry, it takes me to a page with a list of different types of information about the protein. So that's its function, which you can see over here, and then details about the name of the protein, things like synonyms, and you can see some of them in this example here. Here we've got one gene name is CHUK, Chuck, but it's also been known in the past, or by some groups, as IKKA or TCF16. And then subcellular location, where might it be found in the cell, diseases it's associated with, variants in this particular protein in the gene, post-translational modifications or other kinds of processing, where it's expressed, and so on. If we look at one of these types of information, it so happens that this protein is an enzyme. It's a kinase. And one of the kinds of information which is both imported automatically, but also added manually by curators to entries, is the details of the catalytic activity of an enzyme. So this would include things like EC numbers, which are obtained from other databases like RIA. And then there are publications which were used to assign this activity. So in this case, there are 3 publications which support the assertion that this is a kinase. This shows you another example of the kind of information that's in UniProt. This rather pretty cartoon of an imaginary mammalian or eukaryotic cell shows you where you might find the protein inside, together with publications that support the localization to the cytoplasm or the nucleus in this case. The publications which are associated with entries, most of them, as I've said, have been added by a curator manually. Others are imported into the UniProt database automatically. So this is an example here in this green box. We have one paper, this one shown highlighted on the left, has been mapped by a curator. The assertions made in this paper have been incorporated into the entry for this protein. But there are 24 others which have been mapped automatically. So this is data that's pulled in various ways by the UniProt automatic annotation pipelines. Yet another type of data that's included in UniProt is protein-protein interaction. And this slide summarizes the sources of these kinds of data. So there's several different sources, as you can see, BioGrid, the complex portal, which is actually maintained here at EMBL-EBI, and so on, and as is Intact. And in addition to this kind of information, there are cross-references to many other specific database sources for other kinds of data for proteins. Currently about 180. So you can dig into that and find other information about your protein of interest. Moving on to another example of what can be done with UniProt. Once you found a protein entry that you're interested in, and you want to know about its sequences and its features within those sequences, I'll take you through some of those. This is one of the possibilities for obtaining sequence. So the data that we hold is under constant review. We have both curators who are adding data to reviewed entries, and also a very dedicated group of back-end scientists and engineers, so data engineers, who add up-to-date information on proteins in the database. So one of the things I'm going to say a bit about now in the example going on is the viral spike glycoprotein from the SARS virus, and about binding to the human protein ACE2. So this is one of the ways of viewing an entry in UniProt. You may have noticed here there are these links or tabs across the top of an entry. So every entry will have these kinds of links. There's the entry itself, and then there's something called the feature viewer. And this is the view that you'll see for an entry if you click on this particular tab. And in this example, it's the spike glycoprotein from the SARS-CoV-2 virus. And down the side here, you can see a range of different features, domains within the protein, the way the protein's processed, structural features, and so on. In this example, what I've done is I have focused on one particular domain of the protein, which corresponds to the receptor binding motif. So this yellow bar going down the middle of this image represents the region of the protein that's involved in binding to its receptor on human cells. And through the magic of the way the data is linked together within UniProt, in the bottom right corner here, what we have is one of the various X-ray crystallographic or other structures for a protein. And in this case, it's actually derived from cryo-electron microscopy. That's EM, it says down here. You should be able to read that in the bottom of the image. This is the particular PDB entry that this image is derived from. So this is a 3D rendering of the structure, and highlighted in green is this domain here. So this shows you how, if you were to click, if you were using the website live, you could click on any one of these features and they would then automatically appear highlighted in some way in the image of the 3D structure. And of course, one could choose one of these other structures and look at that instead. Going back to the entry view for the spike protein, let's say that we're interested in finding related proteins and we want to do a blast search. So if we now click on that blast button from the previous window, up here we could choose to instead put an accession number in, but in this case, it's automatically, the website's automatically added the sequence from that previous entry into this box here. And so we should now be able to do a blast search. And up front, before you even click go and perform the blast search, you can choose which subset of databases to search, and then restrict that by taxonomy if you wish. And in an example here, this is actually a completely different protein that was used in this example. This was done using a protein called Parkin, PRKN. This is an example of a search that I did recently with this protein sequence. And you can see it's pulled up a lot of quite closely related. If we look at here, we can see 100%, 99.4, and so on, very, very closely related. And then under the organism column heading, you can see, well, that makes sense because these other organisms that have been hit in this search are they're primates. There's chimpanzee, gorilla, and so on. And what I could choose to do now is I could choose to filter these. I could choose to only look at the reviewed entries. As you can see, most of these are unreviewed. They've got the silver grey icon. And the one at the top, the human one, is a reviewed entry. Or I could choose to look at a different species, and you can see just falling off the bottom of this image, there's some entries from rat as well as some from human. So I could have done the taxonomy selection retrospectively at this stage as well. And once I've got the results of a blast search like that, I could then choose to look at the alignments in detail. And you can see here, this is an example of two of the proteins from a blast search. There's the query and then one of the hits, and it shows automatically some of the similarities between the query and the hit result. And it's possible to run multiple blast jobs simultaneously if you wish. You're not limited to just one at a time. There are filters that allow you to filter the blast results, not just by taxonomy, but things like sequence length and whether that sequence has other related sequences in the database. And having got the results of a blast search, and you want to see more detail in an alignment between two or more sequences, what you can choose to do is to use another tool that's built into UniProt called Clustal. It's Clustal Omega is the tool that's used by default. And in this window, you can click to select multiple sequences. And this will help you to find similar regions between proteins, the most conserved amino acid residues, and perhaps indicate functional relationships, evolutionary relationships between your proteins of interest. And this is another way of setting up an alignment if you already know a protein, or if you could choose to paste in accession numbers, or you could paste in a sequence into this search box. So you can do alignment separately from the blast search. Once you've got back the results of your alignment, you can customize the kinds of things that the alignment shows. You can request particular features to be highlighted. And there are two different ways that you can view this this sequence alignment in the UniProt database. You can see it wrapped like this, where you see the whole protein in a series of chunks wrapped around the screen. So we can see here we've got amino acids 1 to 46, and then in the next wrap going down the page, we've got up to amino acid 85, and so on, and it would carry on down the page. Whereas if you wanted, the other option is called the overview, and this shows you a more coarse helicopter view of the alignment and shows you the whole alignment across the window. And if we drill down into some of the detail, some of the things that one can choose to highlight, if you wish, you can choose a particular known feature in a protein of interest, perhaps in your query. Are there known active sites and so on, and known domains? Moving on to another example now. I mentioned proteomes at the beginning. And I said that it basically is a description of the complete set of proteins that an organism expresses. And this is a much abbreviated tree of life, the whole 4 super kingdoms of eukaryotes, bacteria, archaea, and viruses. Now, within UniProt, there's over 160,000 proteomes, and each one of these sets of proteins for a particular organism has its own unique identifier. And in order to construct these, the species needs to have been completely sequenced. And this is expanding. Although it says over 160,000 here, it's continuing to increase, and there are more proteomes which are going to be added in future as other sources of genome sequences appear in the databases. So, for example, at the Sanger Center next door to us, there's the project called the Darwin Tree of Life project, and they are sequencing new genomes, and ultimately the proteins derived from those genomes are going to be finding their way into UniProt. Now, you can retrieve a whole proteome either by searching by identifier in UniProt, or you can go to a specific part of the database that deals with proteomes. You can browse all the different proteomes, and then you can navigate to a particular proteome by using search filters. One of the things about these sets of proteins, they aren't necessarily, although they represent all the proteins known from a sequenced genome, they aren't necessarily all manually reviewed entries. There'll be a mixture of reviewed and unreviewed entries, and for some organisms, there's very little reviewed data. It varies enormously from one organism to another. So obviously organisms like human, mouse, rat, where there's an established community of researchers who've worked on those for decades and have done lots of studies of individual proteins. Hence, there are now massive numbers of curated entries for some of those organisms. There are others on this image which will have far fewer, perhaps even none, annotated and reviewed. So the other final part of the presentation that's relevant to probably to many of you is being able to query the UniProt database for information about disease, or if you're working on a model organism, perhaps to look for mutagenesis data. And you can do this in a number of different ways. There's this link here called diseases, which I think actually in the latest version of the database may look subtly different. I think it might be called diseases and variants. So don't be surprised if this looks slightly different if you would go to our website. An example, if I was to choose a particular disease, let's say I typed Parkinson's or park into the search window on the home page, this might be what I would see. There are numerous genes associated with causing variants in those genes which cause Parkinson's disease. And these are a couple. You can just see there's one that's just about fallen off the bottom of the image. The gene is known as PARC, and this one PARC2, or the disease classifier. And Parkinson's is a complex disease. There are a number of different known genetic forms. Mostly it's idiopathic. It's not caused by any particular single gene or set of genes. And it's almost certainly a large part of it is environmental. What this does, this view, is allows you to explore the different kinds of protein variants associated with a particular disease. The equivalent for a model organism, instead of disease, they're normally known as phenotypes or abnormal phenotypes. And one way you can access, they get grouped together with disease on the website. And I think in this example, it might be fly. I'm just guessing here which particular entry this is for, but I think it's for a fly protein from the fruit fly Drosophila. And the disruption phenotype is a description of the phenotypes, the traits associated with variants in a particular protein. Some of these could be produced artificially, or they could be that they've arisen spontaneously in a particular organism. And the other possible source of variation that's included in this data are things like knockdown with RNAi, which some of you will have heard of, or morpholinos. They're also a short oligonucleotide which can be used to induce a phenotype commonly used in organisms like zebrafish. So there's data from a range of different model organisms. I've mentioned zebrafish and fruit fly. There's also data from the worm Caenorhabditis elegans and other model organisms included in the UniProt data. Sometimes there's more than just a description of phenotype, and that there's individual residues have been modified, and then phenotypes are associated with those specific changes, and they're known and they are annotated in an entry. And these could be the result of modification of a single amino acid, or they might be several. So I've given you a whistle-stop tour of the UniProt database, and I've hopefully shown you how you could perform a search and then find out functional data associated with the results of those searches and sequences for the particular entries that you find. And then once you've got that result, how to analyze those sequences, and then perhaps if you're interested in the wider set of proteins derived from one particular organism, you can explore the proteome from that organism. And lastly, I've talked a bit about the disease and mutagenesis data that's in UniProt. Along the way, I think I said we normally release data every 8 weeks, so it's continually updated. It's all freely available, and it's available in a range of different formats. You can get simple text. One thing that's not actually listed here, if you want it, you can get data in Microsoft Excel format, as well as formats like XML, or if you're interested purely in sequence, the faster format, or tab delimited text. Almost finally, we always want feedback about our website and how well it's performing for you. So please, when you use the database, when you use the website, there's a couple of ways you can get in to give feedback. There's a little icon here on the right-hand side of the window. And if you have problems, there's also the help up here. So if you experience positive or negative feedback is appreciated. Please let us know how you get on. If you want more help, as in addition to the questions which hopefully we can answer, there are other sources of information on the website. There's the help button. There's frequently asked questions like, what is the canonical sequence, and so on. And then these other little icons I didn't mention earlier on. This little one that looks a bit like a toolbox, that holds the results of your searches. So when you do a blast search, or if you take a list of IDs and enter those and do a search, the results of those searches stay there. As I said, they stay there for 7 days. And then another tool, which I didn't have time to say anything about, but is this little basket tool. This is meant to be a shopping basket. So what that does is it allows you to gather together identifiers for multiple proteins. I think the upper limit might be 10. And then you can bring them together into this shopping basket, save them, and then go on to use them in a subsequent search. So you could do a blast search with multiple entries, or perhaps align them using Clustal. And of course, there's a way of contacting us on the website by clicking on this icon here. Then, if you need more help, there's a whole section of the website that's devoted to further uses of the website. There are tutorials and videos. You can see past webinars and online courses which give you a more in-depth guide to particular parts of the website. And I think that then is almost the end. This is hopefully a complete list of all the people involved in creating this resource. The senior leadership team, the key staff on the 3 different sites at Hinxton in Cambridge in the UK, Washington, DC and Delaware in the USA, Geneva in Switzerland, and somewhere in here you'll see our names. Our funding comes from all these sources. And the last thing I wanted to say before I hand back to Ajay and try to help with your questions is we have, I think he's going to tell you a little bit more about upcoming webinars on other topics.
Okay, thank you, Paul. Thank you for today's webinar, and thanks to Rosanna for typing in few answers. So yeah, so as Paul was showing you that slide with upcoming webinars. So please go to our website. You will find these upcoming webinars, and there's actually another webinar lined up from UniProt. It's about uncovering protein function that you can do with UniProt, and that's happening on 17th of November. So yeah, please register for that. And there's another interesting webinar happening in 2 weeks' time. It's about improving data reproducibility for chemosensitivity assays. So if you are involved in designing experiments which involves chemosensitivity measurements, please join this webinar. And as I told you earlier, we record all of our webinars, and all the previous webinars are available on our website as recorded webinars. You can follow this link. So I'll share this slide with you as well later by email as well. So you can actually follow this link and go to all of our previous webinars and find one that interests you. Okay, so with that, we will open for questions. So we already have a few questions, Paul, for you. Some of them have been answered by Rosanna as well. So I'll start with the first question. Is about primary protein sequences. So why is there more than one primary protein sequence for one species?
Why is there more than one primary? Sorry, could you repeat that?
Primary protein sequences for one species.
Sometimes that could be a result of, well, it depends on the circumstance. If it's a curated sequence entry, there could be multiple isoforms for a particular protein. That's one possibility. But if they're not, if it's not a curated entry, it could be that the automated annotation tries to remove redundancy. So any pair of sequences which are completely identical are merged automatically. But there may be a few differences between a pair of sequences, which means that they overlap partially, or one of them is a partial sequence. So there could be multiple entries, unreviewed entries for one particular protein. Does that answer that? I think it does.
Thank you. And the next question is about, I think, the difference in the updated website. So the question is, why does the updated UniProt database not include insertion sequences in mammalian primary sequences of SPB like the original?
Oh, that's slightly too specific an example. I'm afraid I don't know. That's one I don't know. We can ask our colleagues for that one.
Yeah, but this is where I think, as Paul mentioned, that it's a new website and we also require your feedback. So this is a good question for us, I think. And the next question, I think this question is not complete, but I will probably try to modify it. So if someone is asking if the function of a protein is not known, then is there something we can use in UniProt to predict function of a protein?
Again, this depends on context, but there are a number of different possibilities. One is there might be automatic annotation. So most UniProt entries now, a very large proportion, have a predicted structure. So there's an AlphaFold structure. There might also be automatic annotation. There might be domain annotations that have been added automatically, which have derived from databases like Interpro or similar kinds of databases. So I think it's unusual to find an entry which has absolutely no cross-reference information, which could give clues.
Okay, thank you. And I think for this question, I guess you can also attend the webinar, which is happening on 17th November, which will be focused more on function of protein.
Yeah.
And do with UniProt. And there are two other questions which have similar themes. I'll try to just summarize that question. So it's about the protein sequence data that you get from mass spec. So once you get the data, but there are sequences where people don't know the protein, what protein it is, so unknown protein. So are there tools in UniProt where you can put those sequences and try to find matching proteins?
Yeah, the short answer is yes, there are. I have to say I haven't tried doing that myself, but there are people in the team who have that kind of background, who worked, for example, some of the curators have worked in mass spec themselves in the lab. So I'm sure that they would be able to give a better, more detailed answer.
Okay. Thank you. And the next question is, I think somebody missed one of the slide probably, which you showed. So it's asking, can you please show again that how can we find out what amino acid sequence of a protein translates into certain domain of protein?
Yeah, that's the entry viewer version of, feature viewer of an entry. So, this is the coronavirus spike protein. If you choose the domains and sites drop-down menu, you would be able to see different regions and domains within the protein if you were to click on some of these bars here.
Okay, thank you. Now there's again another question. I think is more of a feedback about features in the UniProt website. So why is it not possible to have amino acid sequences in CSV format in a straightforward manner?
I don't think CSV is one of the options, but there's a plain text and there's also tab delimited text, which are almost the same. That would give you something that could be viewed in a flat file, or imported into something that can handle plain text.
Next question is, can you explain how UniProt is related to AlphaFold DeepMind project? Those 3D structures comes. It's the question if those 3D structures come from AlphaFold too.
There are both 3D predicted structures from AlphaFold 2, definitely, but also a large number of structures which are experimentally determined, and they come from PDB.
Okay, so the one question is, how do I explore the target protein for a disease for validation study, if at all this is possible?
If you've got an identifier, a name for the protein, or the name of the disease, then either of those would be a way in to start a validation study. So you could start from either direction, either from a disease name or a protein name, some kind of identifier, or even the sequence of the protein. And that would be an entry point, any of those.
Okay, thanks a lot. Thank you, everyone.